With just 1% difference, the human and chimpanzee protein coding genomes are remarkably similar. Knowing the biological characteristics that separate us humans from other creatures and, in fact, make us human, has been the subject of an interesting and controversial research. In this review, researchers have developed a new approach to determine human-specific adaptive changes in how genes are regulated in the brain. The results of this research have opened a new perspective in the process of human evolution, developmental biology and neuroscience. For a long time, researchers have proposed the hypothesis that what separates humans from their chimpanzee relatives is not only the DNA sequence, but also the arrangement of genes is considered one of the most important factors of difference, meaning when, where and with what intensity a gene is expressed. However, pinpointing the regulatory elements that act as gene attenuators is a challenging task that researchers have so far been unable to tackle. To be able to answer such daunting questions, it is necessary to be able to identify parts of the genome that have undergone so-called positive selection, because the mechanistic perspective provides function of genes. Most of the regulatory elements in the human brain have been positively selected. More than 100 scientists in 10 major research institutes of the world, by studying 232 million pairs of components of human chromosome number 2, showed that this chromosome was created from the fusion of 2 chimpanzee chromosomes. The results of their research have been published in an article in issue 434 of the prestigious scientific journal Nature. The results of this article show that humans and chimpanzees had common ancestors. Humans and chimpanzees are genetically very close. After completing the human gene map, the chimpanzee gene map was completed by scientists. A research showed that 2003 genes are similar between the 2 chromosomes, and the genes that are certainly similar are the genes related to the ability to develop speech, which has developed very quickly in humans. The genetic difference between humans and chimpanzees is related to 35 million single nucleotides, 5 million deletions and creations in chromosomes and new chromosomal combinations. The similarity of human and chimpanzee proteins is on average in 2 amino acids. About 33% of human proteins are sequenced similar to chimpanzee proteins. Duplication and copying of a small part of chromosomes is the main reason for the genetic difference between humans and chimpanzees, and 98% of the genetics of these 2 creatures are similar. About 1 to 2 million years ago, the common ancestor of humans and chimpanzees diverged from each other. All apes except humans have 24 pairs of chromosomes, and humans have 2 less. Therefore, the ancestors of this family had 24 chromosomes, and in humans, 2 of the chromosomes have merged, and humans have 46 chromosomes instead of 48.